And then share screen. So, sorry kayo, nakalimot ko sa ako ang online. Okay, so we are just having a review on the history, you know. As I have said, last time, it is where, tanawan na akong PowerPoint, anong nakuan. Isa nga kay na something wrong. Okay na. So ito lang, no? I hope na nabasa niyo yung PowerPoint presentation. So 2 million years ago, kanim na mention kanina, the golden, silver, bronze age and iron age. So sa iron age, it is where like iba no sa salida sa una katong gira-gira pa gamit sila og kanang iron. So this was also the time na agriculture we started using iron instead of wood or stone. And agriculture in relation to energy civilization. So in this, since we discussed on photosynthesis, by the way, nag-review lang at the moment about our previous lesson last week, highlands. So, as I have said, yung ating agriculture, una na ako na ipasat ano maayos ang kanin. So as I have said, agriculture started with civilization. So, kailan nagkamata ang tao, di hapon din nag-start ang agriculture. But as I have said, hunter or gatherers lang sila. No, kanina, sabi, that was 10,000 years ago based on 10 timelines. Ngayon, it could be estimated daw based on the Earth carrying capacity. It is estimated na 20 to 30 million ang capacity ng hunters or gathering sa Earth. Okay, set up cocktails. Now, and then with the kind, we all know that sa photosynthesis na topic, naman we all know that we depend on sun as our solar energy or source of energy. No, not only plants but also animals. Kasi as human beings, kailangan po nato ang sunlight, no? And what will happen to us kung wala na sunlight? I mean to us, kasi to plants, they will all die, of course. Morning. And but to us, magkaroon pa ng sakit, di ba? Kasi meron tayong vitamin D in our skin to be activated by sunlight para it's also for our bones, atong system sa skin. So in plants, we literally die or they literally die and we will also die out of scarcity kasi we always depend on plants. Our animals depend on plants and we depend on both plants and animals. So this is so much to discuss to you kasi we have done this already. Ipakita lang ako sa inyo kung saan makita ninyo dari. About how we transition from hunting, gathering to agriculture. So, meron din tayong tinatawag na Pleistocene age, Neolithic of the present day. So this based on human population, no? So ibig sabihin with the present day, ang percent nato sa population, pila na data karon tanan in the Philippines, mga Filipinos around the world. Pila pa tanan? 80 million? Kami na 10 million lang. Ha? Updated na inyo, ha? Ano, close din ako ang Google. Ang US pa dyan. Ano sa money? 2022, 4 million? Sa Amerika lang siya?
109 million, ma'am.
Nakay computation dara? 112 million Filipinos doon na karon. How about all persons in the world? Ha? Sige daw, paki-search niya mas effective na yung mga kuan. Okay, sa unang po, padaghan na, sige sir, kuan ha, 7.9 billion. So, hello dear, sorry kaayo, nag-repeat ako. So, 7.9 billion. O gamay-gamay pa man. Around the world, population, no? So, but sa una kasi, dati, ang stars daw, if gamay ra kag anak, so, kubre ka, hiding. So, finally, makasayo na yun si Ivy karon. So, kasi sa una, mas dagan kag anak, mas asset. Kasi wide kaya ang area, but walay maging farmers, diba? Walay mag-farm. Kasi the only work we have is farming. But ngayon, dagan kay kaganak, maghilak ka, no? So everything changed na. So it is not actually based on the ignorance on how to grow plants, but the technology itself, no? And support. Alam na natin yan. So, i-rundown lang na ako kasi wala ko na kanina mention last time. So, from hunting to gathering, daghan pa kayong seeds, no? Available. And, again, 10,000 years ago, na-develop na ng tao during, there's also certain theory na after the last ice age, so like yung panahon ng mga dinosaurs kay wari. That's just a theory, ha? Kasi as I have told you last time, yung geology is they call it the fake science kasi based lang sa mga haka-haka, like marites lang primitive ba na uy, kanang kaila ka ani, ba? Kabalo ka kung saan nahitabo last time, Nantod ang mga nakalap na mga balita, so they put it into a publication, so naging yan, which still always need to be kanang verified. So 10,000 years ago daw, after daw sa last ice age, so na-melt ang na-melt ang mga ice, so diha nag chance na makagrow na ang plants. And nag-start na po ang agriculture, and that was 10,000 years ago. So nawala na po ng mga dinosaur. So, as I have said, na-mention ni kanina last time, ang fertile crescent, ang magkita ko ang ano, for sure. So, diha sa Mesopotamia or now, Iran, Iraq, no? Or in Egypt. So, here specifically, Iran, Iraq. So, this is the fertile crescent. Diha nag-start na civilization, no? Kasi mahilig sila mag-record ng mga bagay-bagay isulat sa stone, no? Like kanang, pero kita kasi mga Pinoy, hangtod karon nakasulat yapon sa bumbo ang mga pangalan nato sa boarding house. So, dapat kabalot ta how to put it into writing ang mga thesis book nato diha ni Babing, no? Shema, kasi ito yung research natin. Augustine. Okay, so para makita pagyapon sa mga coming years, ang katawalay ka Carlo. Isa tayong study sa una, Carlo. Nag-finish ka memorize. Ang sa nga bacteria, ay kuan mo ka, di ba? Ito? Krebs Cycle. Is him advisor? Si Sir Tidoki. Okay, so, ano nakalimut ako? Nako. So, yeah, and also from some parts in the world, like in Ecuador, kasi tropical sila. So, most of the plants came from there. Ayan. And China, where rice came from. And sabi nga, last time do, na also, actually, gikan sa Vietnam and then giangkat sa rice. Then, giangkat sa China and then later on, ang China na ang nag-develop sa Yangtze River. And these parts of the world, the Mesoamerica, the Mexico, Guatemala, Belize, Honduras, and El Salvador, these are the Mesoamerica or 5 countries na nag-start ang mga crop production. So, this is where they, Mexico, Honduras, and Panama. Kasi tropical sila. And pati ang ang sana, drugs po na develop ang weeds. So, and then part of the transition also is a continuous na production sa ating fertile crescent and slow ang development process talaga. So, also, na-mention dito ang vegetaculture region where dating mga, actually in some other sources, ang first na mga farmers were actually women than men kasi most men were fishermen. So, ang mga babae mo ay nabiling sa balay mo ay nag-garden. Have you, and there were like retinoids, no? Kasi first practice ng agriculture talaga is fishing. So, and una yun kasi than even animal production, no? But na-mention na natin last time, no, this is for crops, but for agriculture last time, ang unang na domesticate na animals, ano nga yun? Dogs, cats, rats, sheep, cattle, ox, and pig or swine. And sa vegetable naman, we have sweet potato, yam, taro. So those were fiber. And then in southern Mexico, we have corn, maize. And bean, vines, So, kani detail kasi niya kung di unsa pagka-develop sa mga varieties. No? Which, ato na lang ng, and also this one, how seed culture originated, mga 8 to 10, probably 8 to 10,000 years ago in the Tigris and Euphrates River. Also, the fertile crescent again, and in the Indus River at the same time sa northern India and Pakistan, and Wangho River sa Yangtze, northern in China. So, kita na mo ani nga mga places sa pictures lang. No? Isa ta magdali. Kay pandemic pa ba naman? Isod pa mag-adult play. So, irrigation also is part of our history. No? And wet dry season ng mga cereal grains. Yan. Ito yung mga ancestral wheat and barley before. So fast track natin. From the flat, the ground, it moves to the highlands and valleys, and then the development and irrigation, tillage, and mga selection ng varieties such as beans, navy, kidney, lima, and peanuts. So, very long talaga ang history in terms of agriculture, which is again, Sharon, di makalimtan mo na yung 10,000 years ago, balik-balik. No? So, it's because also of agriculture, because it's our source of food, so it's where civilization developed. So, in the Near East, Far East Asia, Mexico, and Peru. So, along the way, na-change na rin yung ating based sa ang ating food preference based sa ating history, sa ating diet, no, which is composed mainly ng carbohydrates, proteins, and vitamins. So, like for America, kaya nga hanggang ngayon yung kanilang staple food ay wheat, diba? Sa kanila galing yung talagang maize and peanuts. Sa Africa naman, sorghum and soybeans, and Middle East, wheat, barley, and beans. So, and Asia, rice and soybeans. So, ayan, ang mga origin ng major crops and their country. But sa ating vegetable, last time, na-discuss na po na nato, diba? Asagikan. So, asagan ikan tong Brussels sprout. Sa mga country. Kung sa ng Brussels sprout, sa ito scientific name, agoy, mag-review na maka-scientific name, ani Madam Bim. Bali makagrupo, Madam Bim. Ha? So, from the word Brussels, no, Belgium, kinasagitan. So, imagine na lang ninyo, no, basta more on tropical fruits. So, tropical countries would ang gikanan, anak. But coconut, tanawa sa South America siya, origin. But as I have said last time, those are all arbitrary. No, mga theory lang na siya, maritest lang na. Kasi based on hearsays lang, not really actual evidence. Yeah. But yung okra, medyo nag-dry siya, which is from Africa. And as I have said, ang ating Philippine hemp, yung ating abaca, is really an indigenous native in Philippines. It's only from Philippines. Yes, sir. And, sorry lang, abaca. And banana, Southeast Asia, even citrus, ang mango, no? India, Asia, papaya, of course, and pineapple are from America. Chrysanthemum is from China kasi baka maano na. And yun, orchids, China. And mga centers of agriculture natin, we have Western Asia, Egypt, and Europe. O asa na ang Asia? Ang Africa, through the Nile River siya. Southern Asia also, of course, Central Asia and Eastern Asia. So, tanan man dahil gihapon na, ma'am. Yes. In China, Thailand, Malaysia, Indonesia, and Philippines. That's why ang staple food natin is rice. And Southeast Asia. Oceania also. So, ilaman mo asa ang Oceania, diba? Sa Pacific Islands. So, sa kanila, Taro, it's where I came from. Ay, dili ko kuan ha. Dili ko Oceanian. Pero ang ato ba yung parang kaninong inuan, di ba? So, gani, Indo-Malaysian. Ha? In-check. Sana ko lang, no? Kanang Japanese. Ang sakit na. Sa una, naghilak-hilak ang inyong atong mga lola kung ma-rapean ang Japanese. Karon pati natulip paya kay na-rapean sila Japanese, no? Kasi tayo. Makadala Japan. Anyways, so ana lang, no? Para-paraan lang ta, mga Pinoy. So, as I have said, sa Ilaha, sa Samoa, where I came from before nag-RMC ko, is they have taro yams as their staple food. And even breadfruit, no? Sa ato, isa lang ka-variety ang breadfruit na to, but sa Ilaha, they have at least 4 nga gina-consume. And the rest are parang backyard na lang. Pareho sa okra, gi-backyard na nato na diri, diba? Pero adto og kanang was that Vietnam or Thailand? Ay, sorry. Ha? Nga ilahan ng plantation ba? Kasi that's anti-cancer. No? Ang okra. So, kit-kit din mo okra kay ang okra di ba mangundang bunga for 3 months. 4 months. So then America. So it's the first center of the world of agriculture. They claim it to be, but in America, but as na-mention naman diba nga, natin mga sa Fertile Crescent or katong Mesopotamia or Iran, Iraq, mo yung kinauhan based on historical facts, no? Sa mga sulat-sulat na record. Kasi nga kita, most probably, ang Pilipinas nga, sabi ko nga last time, diba? Nag-exist na ang atong mga ninuno before pa na-discover ni Magellan. Kaso wala lang pa nag-record. No, ang yung sa mga part sa history nga, imbis mag-record tao ng mga unsay nahitabo. Sige naman tong bandal, no? Okay. Kasi in psychology, ang vandalism is a form of kanang psychological problem. Hindi siguro ma-express sa sarili. Sa dahon, gisulat ang oo, no? Sa una, no? Ano, dugay kita mahuman. Telegrama pag sa una, diba? Tapos wala, unlike other countries, wala ta atong ganing papel nga gigamit ni Jose Rizal, no? Nga kanang gisulat. So anyway, dinan nato na sila sisihon, no? Pero karon mag-publish na kita sa atong mga, diba, papers, diba? Kung assign mo sa research, kasi nasa sa research na rin. Trabaho sa research. Wala. Sir Pundal. Kami na kung manager na ka sa research ninyo. No? Si Sir Carlo. Of course, Sir Clark. Bagtik ba yung nag-advisor? So. Takdan po ka eh. So, Southern Mexico is the center naman among the American countries. But, and also their domesticated major crops are sweet potato, cassava, peanut, gourds, pineapple, tobacco, rice, beans, and cotton. So, hinay-hinay lang yun nato o ng review. So, these are the global centers of crop of origin in the map kanang naka-red. Nawala na po ang ako. Ayan. Ayan. Southern Mexico and Central Peru, Bolivia, Chile, Brazil, and Paraguay. In the Mediterranean, Near East, so na dari ang Mesopotamia, Central Asia, India, China, and Indo-Malaysia. Philippines. And yeah. So dito, you can see most probably where the different crops began, domesticated. And in the Philippines kasi, since we were colonized by a lot, not a lot talaga, tulo lang man. Spain for 300 years, Japanese for 3 lang, no? Ilang ka years ang Japanese? 3 years or ano? Something 3 kasi 30 years. And ang American. So sila lang ang medyo nakakuan sa tuang. Nagdala sila og mga crafts, no? Like bread from started siya sa English, sa American and Spanish. And so yeah, so inyuha lang nang i-familiarize. Of course, ang rice sa China, gano'n siya. Most are arbitrary or kanang ano siya, kanang sapaw-sapaw, no? Kasi iba-iba ang claims. So another map to show you by center or point of area. So ito naman ginalist down niya talaga sa how did soybean originate, no, from Asia, China, Japan to US. So hindi na natin yan isa-isahan as also sorghum kasi and maize and rice areas. So but now rice, the top producer of rice is what country is the top producer of rice? Is it Thailand? Yes, Sir Ron? It's Vietnam, di ba? Ang top producer. Ay, sorry. So, ayan. So, lastly, they said that the word bigamis or negritos from Central Asia are not agriculturists, and food hunting, fishing, and gathering wild fruits as our source of food, and we call it fruit agriculture or kaingin or shifting cultivation, or the slash and burn or swell and light. So, ito yung pinaka-start ng ating agriculture. Before, where nagkaroon tayo ng industry na agriculture sa pre-Spanish Philippines. And after that, of course, as I have said, very slow ang ating history. So, we have spent so much sa ating review, but kasi wala to na ako siya na-discuss sa inyo. And our classification, you still remember? Hindi siya mao. Nako bagong Last time, I forgot the ornamental classification, di ba? Kaya wala na ko nakita. Tama ba? But na-send na ko sa inyo, right? And then we talked about branches and wait lang kasi wala dito sa akong ESP. mamaya na lang ako i-review. Then let's proceed with okay. So let's proceed with the respiration. Kasi we've done the also the plant anatomy last time and ano ba? Plant anatomy and functions, diba? So daghan kay tag na take up last time. And to continue, and of course respiration, ka-remember pa man mo no last time? And to continue that, let's have cellular respiration.
Welcome to our video series on cellular respiration. Before we get started, I'm going to ask you to do a few things. Don't be a passive learner. Have paper and pencil handy. Pause the video frequently. Rewind and rewatch. Draw your own pictures, because as you know, pictures are your friends. And ask questions.
Hi, it's Mr.
Hinay kasi kayo siya. So, kanina lang isa ka video.
Anderson. In this podcast, I'm going to talk about cellular respiration. Sometimes students confuse respiration, breathing, just breathing in, and cellular respiration, and they are linked. Cellular respiration, however, is going to take place at the level of the cell, more specifically inside the mitochondria, and so we need oxygen for that. But it's basically taking our food and then breaking it down in the presence of oxygen to make ATP out of it. What if you're a bacteria? Can you do respiration? You sure can. You don't need a mitochondria to do respiration. They can actually use their outer membranes to do aerobic respiration. And so basically, if you are a track athlete, so this is Usain Bolt right here, when you run, you're using respiration to make energy in the form of ATP and allow your muscles to move. And so this is a quick little study I did. I took all of the world records currently right now from the 100 to the 10,000. And so this is Bekele is the guy who owns the 10,000 meter record. And what I did is I figured out their pace. In other words, how fast they're running. This would be in meters per second for the 100, the 200, all the way down to the 10,000 meter run, which is a little over 6 miles. And what you can see in this graph is that the pace is quickly going to drop off, and then it's pretty much going to stabilize. And if we were to go out to the marathon or continue, basically we're pretty effective at running at a specific pace, but we die really quickly when we're sprinting. And so a way to think about that is the two things we're going to talk about in this podcast is we're going to talk about aerobic respiration. So aerobic respiration is going to be respiration in the presence of oxygen, but we also have almost like a turbo button that is anaerobic respiration. And so if we really need to go fast, we can get that extra speed that we have up here by doing our anaerobic respiration. The problem, if you've ever run, is that when you get out to this 400 meter, you get a buildup of lactic acid, and it's incredibly painful, and so you can't keep that pace going. Example of a lab I did in class was, and it's weird how this exactly mirrors it, what we did was the muscle fatigue lab. So basically you had a tennis ball, and in one hand you had to squeeze it as many times as you could in 10 seconds, and then do another 10 seconds, and do another 10 seconds, and this is the class average. So the class average was, it looks like around 25 times in 10 seconds. But you can see that it quickly drops off, and then it kind of levels off. And so the same thing, this would be that aerobic respiration, and then this is going to be that anaerobic respiration. And it was fun to see the students' faces, because as they start to go anaerobic, your arm just starts to build up if you have lactic acid on the inside of it. But before we get there, let's talk about respiration and what it's for. It's for heterotrophs. So we're heterotrophs, and basically what we're doing is we're taking organic compounds in the presence of oxygen, and we're converting that to carbon dioxide and water. What else are we generating? ATP. Now, what kind of things are doing this? Animals, fungi, bacteria are all heterotrophs, and they're using the organic material to actually make energy. Luckily, we have autotrophs like plants and algae, and basically what they're doing is they're converting that carbon dioxide and water back into organic materials. The only thing that's a little deceptive is that plants are also going to break down those organic compounds, and so they do cellular respiration as well. And so everything's doing cellular respiration. It's how we get energy out of our food. Okay, here's our equation. And again, if you know what photosynthesis is, this is simply the opposite. We're going to take glucose in the presence of oxygen. So here's glucose and here's O2. And then we're going to break that into carbon dioxide, water, and then we're going to generate a little bit of ATP. Now, where is the energy sit? The energy sits right here in this hydrogen on the outside of that glucose. And watch what happens to that hydrogen. It's going to fall down and it's going to grab onto the oxygen because oxygen wants electrons. And so that's where the energy is coming from. And what the energy is used to do is it's used to make ATP. And ATP is that little fuel that we use in all of our cells. This slide is funny, but it's saying this: behold the power of oxygen. So this fire comes from oxygen pulling electrons close to it. And so there's a huge amount of energy found inside that pull of electrons towards oxygen. Now, if we were to do this inside our body, we could get a lot of energy out of our food, but we would also burst into flames. And so we do it in a really controlled process. Just like when we learned photosynthesis and we had to learn the parts of the chloroplast, when you're learning the cellular respiration, you have to learn a few parts of the mitochondria. So first of all, we have these folds on the inside of the mitochondria. Those are called the Cristae. And basically what we have is two membranes. We're going to have an inner membrane right here, and then we're going to have an outer membrane right here. And then this space in the middle is called the inner membrane space. And on the inside, mitochondria, we think, used to be bacteria of their own. And so they'll reproduce through binary fission. They have their own DNA. They have their ribosomes. But they are kind of almost living inside us, not as a parasite, but as a symbiont. They're actually helping us as we generate energy. So there are 3 steps in cellular respiration. Let's start with the first one. So the first one is going to be glycolysis. The second one normally we think of as the Krebs cycle. And then the third one is going to be the electron transport chain. And so the first one, I love this diagram here because it's putting glycolysis outside the mitochondria. And so this is going to take place outside the mitochondria. Where would that be? Well, that would be in the cytoplasm of a cell inside your body, or it would be right outside of bacteria. But what happens in glycolysis? Basically, we're taking glucose. Glucose is a 6-carbon molecule. And in glycolysis, we're going to break that down into 2 molecules of pyruvate. Each of those have 3 carbons inside it. So the 2 3-carbon molecule, that's what glycolysis does. What do we generate in there? Well, we generate a little bit of ATP. For 1 glucose molecule in glycolysis, we're going to make 2 ATP. The other thing that we make is a chemical called NAD. What we're basically doing is we're transferring high-energy electrons to NAD, and we're adding protons to it as well. And we'll get to NAD in just a little bit. Let's follow pyruvate then. Pyruvate is going to diffuse into the mitochondria, and then we're going to have this Pyruvate Dehydrogenase complex, and basically what it's going to do is it's going to convert that 3-carbon molecule into acetyl-CoA. This is coenzyme A. So basically now we have a 2-carbon molecule. That is going to go into the Krebs cycle. Now, since we're going from a 3-carbon pyruvate to a 2-carbon acetyl-CoA, we're giving off carbon. And that carbon is going to be given off in the form of carbon dioxide. And so when you breathe out, a third of that carbon coming out of you is going to come right here from this complex inside the matrix, we call this. I should have said that before. This is the matrix on the inside of the mitochondria. Okay, let's keep watching acetyl-CoA. So it's a 2-carbon molecule. Where does it go next? It goes to the Krebs cycle. And so in the Krebs cycle, we're going to break it down further, and we're going to get rid of these 2 carbons in acetyl-CoA, and we're going to give those off as carbon dioxide. So we're getting rid of carbon dioxide. What else are we producing in the Krebs cycle? You can see here that we're producing a little bit of ATP, 2 ATP, but we're also adding energy, again, to NAD. And we're adding energy to its friend, we'll call this FAD. And so what do NAD and FAD have? They have these high energy electrons, and they're going to carry those electrons to the 3rd step, which is the electron transport chain. Okay, let's get to the electron transport chain then. All of our energy pretty much that was in glucose is now in NAD and FAD. So they're going to transfer their electrons, and those electrons are going to go through what's called an electron transport chain. Basically, they're moving through a series of proteins, and the energy of those proteins is used to pump protons. Protons are going to be hydrogen ions to the outside of this inner membrane, into what's called the inner membrane space. So now we've built up all of these protons right here. What happens to the electron? The electron is going to be added to other protons and oxygen that we breathe in, and that's going to make our byproduct, which is going to be water. And so let's slow that for just a second. The oxygen that you breathe is moving in here, and it's going to be that last electron acceptor right in here in the matrix, and we're going to take the protons. What happened to those protons? They'll actually flow through a protein called ATP synthase. Those protons will combine with the electrons and the oxygen, and it's going to make water, which is going to be a byproduct of that. Now, how much ATP do we make down here? Well, we can make around 32 or 34 ATP in this last step. And so in the electron transport chain, we're making a whole heck of a lot of energy. And so it's worth taking a look at how that actually works. So let's go to the electron transport chain. So kind of to situate ourselves, what do we have? Well, we have NAD, our friends NAD and FAD. What are NAD and FAD passing off? They're electrons. Those electrons are going to move through the electron transport chain like this. Every time they go through one of these proteins, it's going to pump another proton ion out, because that's the other thing that NAD and FAD are bringing. They're bringing these hydrogen ions. So we're going to pump these ions out here, and pretty soon what you get is a heck of a lot of positive charge out here in this inner membrane space. There's no place for it to go. In other words, every NAD that we drop off, we're going to move these electrons down, and we're going to generate a whole heck of a lot of positive charge in this inner membrane space. Now, if you look right here, FAD is actually dropping its electron a little bit farther down, so it can't generate as much, but we're pushing out either 3 protons or 2 protons, depending on if it's NAD or FAD. Okay, what happens to all of these protons out here? They can't go anywhere. They can't go outside the mitochondria, they can't come inside the matrix, but they can move through this. This is called ATP synthase. That's the name of this protein right here. And basically, this is the site of ATP synthesis. And so basically, as every proton flows through it, every proton that comes through, we're going to generate ATP. And it almost works like a little rotor, that every time a proton goes through, it switches it and it attaches that phosphate onto ADP to make ATP. And that's why in the electron transport chain we can make all of that ATP. There's nothing special about it. It's just that we're storing all that energy, and instead of releasing it in a ball of fire, we're releasing it in little bits to make a heck of a lot of HTP or ATP. Okay, so there's a problem. What happens if you don't have oxygen pulling that electron the whole way, or let's say you don't have mitochondria present? Well, then you have a problem, and the problem is this. It's okay to take glucose during glycolysis and break it into two pyruvates, because you're going to make a little bit of ATP, but the problem is that you're adding those electrons to NAD. And so basically what's happening is that we're adding electrons to NAD plus, and we're transferring it to NADH, and so pretty much what happens is there's no more of this. And so glycolysis has to shut down. Even though we can make a little bit of ATP with each breakdown of glucose, eventually there's no NADP plus, and so the whole process has to stop. And so nature, of course, has a solution to this, and the first one is called lactic acid fermentation. This takes place in your muscles, especially when your muscles are under a huge amount of stress, like if you're sprinting or if you're holding your breath for a long period of time. And so basically what's going on, again, there's no oxygen, there's no mitochondria, so let's look what happens. Basically, your cells are taking glucose in glycolysis and breaking it down into 2 pyruvate molecules. So we were stuck here, remember, with the NADH. But then there's a further conversion. Basically what you're doing is you're converting that pyruvate down into lactate or lactic acid. The nice thing about that is it's accepting these electrons, so we can make more of this NAD plus, and then this can be recycled again. And so basically what happens is that you can have this process occurring with glucose, over and over and over and over and over again. And every time you do that over and over and over and over again, basically you're making 2 ATP each time. And so if you've ever done sprinting, when you're sprinting, you're getting aerobic respiration, but you're also doing anaerobic respiration on top of that. The problem with that is you're going to build up lactate in your muscles. And that lactate is like a toxin. You're going to have to break it down. And that takes oxygen. And so if you've ever watched a sprinter, especially somebody who's run like the 400-meter dash, when they're done and they're interviewing them, they have a hard time doing an interview because they have to keep breathing to take in more O2 and eventually get rid of that lactate. And so lactic acid fermentation is going to take place in some bacteria and in muscle cells, but we have another solution to this in bacteria, anaerobic problem of stopping right here with this full NADH, and that's called alcoholic fermentation. Alcoholic fermentation works the same way. Basically, we break it down into pyruvate, and then we break that further down into a chemical called ethyl alcohol or ethanol. It's accepting these electrons so it can recycle this NADP plus again. The only difference here is that when we made lactate, that was a 3-carbon molecule. When we do alcoholic fermentation, what we're making is carbon dioxide, and we're giving that off. And so if you were to take yeast and put them in a bottle with a bunch of fruit juice, basically what they'll do is they'll use up all the oxygen, then they'll switch to alcoholic fermentation. What are they going to build up? They're going to build up ethyl alcohol. That's simply how we make wine. And there's also going to build up carbon dioxide, which we could let go. Or if we're making beer, that's the carbonation that we're going to find in beer. And so again, cellular respiration is just a quick way to get energy out of glucose. We use glucose as an example, but we can do cellular respiration on pretty much any type of food. And it's a way that we get energy. And we're doing it, bacteria are doing it, plants are doing it, and I hope that's helpful.
Okay, so it's a more simpler form of explaining to you what does respiration do. Of course, the main idea is to produce what? The ATP, or it's as a source of energy. Pero along the process, it's a very complicated kasi even meron tayong tinatawag na aerobic and anaerobic respiration. Of course, from the word aerobic, with air. So that's the usual fermentation. But without air, like for example, kaya meron tayong tinatawag na alcoholic fermentation or lactic acid fermentation. So, and thanks to that, pwede tayo mag-produce ng ATP without air. So, nanatay wine, no? Mga hubog na pagdion niya. Nanatay ka ng cheese, that source of product of fermentation. And lactic yogurt, diba? Lactic acid bacteria fermentation. But along the way was a complicated process, no? Yung glycolysis. Actually, lahat ng ito ay na-send ko na sa inyo sa GC na mga handouts. So at this point, of course, musugot pa rin si Madam Bean kung di makasayaw si Ivy, no? Dili pwede. So we're gonna have another short quiz. Nasa ako yung taon. Regarding the products of respiration. Kasi in respiration, we have glycolysis, no? And the Calvin cycle. So in glycolysis, it's one process wherein yung ATP ang ating na-produce. And then from glycolysis, we'll go to the Calvin cycle wherein it's usually a cycle, but then ano lang po siya para ma-produce. And all in all na siya total na ma-produce na ATP. Diba? Along the way or along the process. So, there would be like 38 ATP all in all na ma-produce. Sa first step pa lang na siya. And in the end, wala lagi na rin sa kumuha.
32 to 34, ma'am.
Ano yun?
32 to 34 ang last step.
32 to 34. Depende siya, no? Hindi siya ano, pareho. So, but in the board exam, medyo 2 or 3 to questions ang magawas sa respiration and also sa photosynthesis at saka sa mga plant anatomy and their function. So, ready na mo? Ready na mo? Ang inyo halang i-mention ay products of respiration. Isa. Ay, i-flash na ako. Nawala na po ito. Dali lang ha, kaya nawala na sa ko. Annotation. So, and we can now move on after this sa ating prior topic talaga for this day is yung ating plant growth and physiology. So, we're going to talk about plant growth regulators. Pero nga hindi na ako ma-open na rin. Kumana ni Ron. Nara. Cellular respiration. Okay, so you can also mention any lang na makita ninyo nga mga chemicals within respiration. Maski dili pa siya ATP products. So I guess wala na ako nito send no kasi naka-word akong send sa inyo ha. So the main parts ng cellular respiration, which already have mentioned, and then we have the Krebs cycle, and lastly the ETC or the electron transfer chain. So dili naman ako pa isa-isahon sa inyo ha ang steps, but usually yung mga products na mention sa along the way from the process from glycolysis hanggang electron transport chain, like pyruvate, NADH, PEP, ATP, phosphate group, ADP. So example lang na siya sa mga products, no? Even wine, pwede pa siya, no? Mga products ng mga fermentation. Alcohol, lactic acid. So ready na? Ethanol. Ano sa pa? Daghan man siya, diba? Carbon dioxide. Acetyl coenzyme. Kaso medyo lisod siya na i-mention. Citrate, citric acid. So ito na yung Krebs cycle. So sa una memorize ko ni kaya kailangan sa compre. So from acetate coenzyme A ma-transform into citrate and then mag-release ng carbon dioxide, NAD, NADH siya, maging NADH and then another phosphate, ADP, ATP, pero naka ni siya. Yan. So, ito yung sinasabi kong mga na ATP net yield bawat level. So, but don't forget na asa nga cell. So last time we did have discussed the different parts of the cell and their function, di ba? So asa mag-take place ang respiration in the mitochondria. Okay, very good. So specifically yung ating electron transfer change ay nasa sa loob na ng mga membranes ng mitochondria. So na-explain man ito kanina, diba? Ni Anderson. So 38 pa na lahat ang ATP yield. But depende yon if it's eukaryote or prokaryotes. But when we say eukaryotes, those organisms na merong what? Merong membranes, merong membranes ang kanyang cell or nuclear membrane. So, example ay, of course, yung ating mga plants, no? Animals. So, pag prokaryotes ka, naka sa mga bacteria. So, lahat ng eukaryotes will have 36 ATP lahat. Ayan, which is doon siya makukuha no from the glycolysis to ATP for ATP sa ATC dito sa acetate coenzyme A 6 ATP sa Krebs Cycle 18 at sa FADH na 14 4 and 2 lastly. A total of 36 ang sa eukaryotes, but sa prokaryotes are 38 lahat. So, asa ang difference nila? Nasa grooming, I think sa grooming phase, no? Alimot ko ito. Really ay. Sa glycolysis and sa Krebs cycle. Sa glycolysis. So there are, mas dagan sila o ATP ma-produce. Okay, and napaganin different molecules na pwede natin gamitin for respiration. It's not only all carbohydrates or polysaccharide, but also fats. Second is in level na siya ha. Second, fats. So, kung di ka mukha nag-rice, ang fats din mo ang iyahang gamiton. But wala pa dyan kay fats, kay payatot ka, kung sa'yo mahita mo, mahurot mo muscles. Saan na lang mo? Ang atong pagkagwaping dala. So, kasi ang i-reutilize niya ang proteins or amino acids. And then, magpadako pa man tag-muscle balance, kaya atong mga irog dala. So, questions or clarification before tayo mag-quiz? Medyo klaro na ba? So, anything na lang, no, even the phase or the step na ma-mention ninyo basta may kinalaman sa cellular respiration. Okay, para ma-exercise yung tatanan, then we're gonna have individual, no? Muna. So, start tayo. Hindi man mag-open lang kayo yung steps man. Mag-mention na kung anything that talks about respiration. Even, mitochondria ba? Kasi makaunod. No? Okay, so bayong yapon ka na. Kaya, sumakulat ka sa mga kay sir. Any step or product or anything that involves respiration. So like, carbohydrate. Sharon, lagi niya ha, itagaan ka na mo o in photo. Kapangit ko na yun. Asa mas? Ay. Di maklaro, no? Maklaro? Oh, so muna ang full process ha sa glycolysis. Sharon, wag ka mo i-mention, Ana. Okay, start ka na sa direct. Start ka sa mga sir. Okay, people. One, two, or hindi na ka. just a very simple exercise para ang terms is ma-remember niyo ang terms sa respiration. Okay, so at least man lang, no? Kasi no need to really memorize them all. And the key to physiology is to understand them, to know the parts and understand them. So sa photosynthesis, we have understood na it's a process where plants produce carbohydrates and food. So carbohydrates lang ba ang kaya i-produce sa tanom? Isa pa. Proteins. But the proteins, of course, during the during the metabolic processes. Of course, along the way sa, what we call that, sa ating, dito na, sa Krebs Cycle. And when you go back sa photosynthesis kasi, ang main product niya are carbohydrates. And then, along the way sa pag-produce ng ATP, pag-abutin mo sa Krebs Cycle, nanan na siya yung mga byproducts na ma-produce. That includes katong mga phenolic compounds and others. So that's too much chemistry noon. Kanang biochemistry or chemistry. So, but it's very good that we understand their function and the processes involved. And we've learned na sa photosynthesis, there are 2 types or even 3, diba? The what? The C3, the C4, and the CAM. And ang anong difference nila? Of course, C3 yung normal na photosynthesis. So anong meron sa C4 na wala sa C3? Kasi, yes? Yes. Okay. So it's more efficient ang C4 and C5 kasi it has the capability to process photosynthesis without the sunlight already. But in the C4, meron siyang bundle sheet cells called the trans anatomy where it can fix the specific. It can produce glyphosate G, 3G, 4 glucose product, than the C3, the 3 glucose product. So, and the CAM is another instance because it's a carcellation as CAM pala, dili CAM, acid metabolism, wherein it has the ability na mag-conserve of water kapag init na masyado, in sa mga desierto like the cactus. So pag ma-preserve, ang sa iyong buhaton, it will close its stomata during day. And i-open lang niya during night. And so, maka-take in ang carbon, the oxygen, and pwede na siya mag-photosynthesis. So, therefore, ang photosynthesis has the dark and light reaction phase. And eventually, yung product ng ating photosynthesis, which is glucose, mainly glucose lang din siya, but of course, in other form, pwede din siya fructose and so on. Basta carbohydrates lang din siya. And in the respiration process, mag-produce tayo ATP by number pag prokaryotes or eukaryotes siya. So, pag eukaryotes pila ganito, 30. Yes, pag prokaryotes pila ganito, 88. And anong difference nila sa glycolysis? 4 and 6. So, and also pwede rin maging aerobic or non-aerobic respiration. Of course, ang aerobic respiration yun yung normal. Pag abot sa anaerobic respiration, what are the 3 types? 3 ba to? 2 types. Fermentation. We have the
Lactic,
alcoholic and
lactic acid,
acid fermentation. So, alcoholic fermentation, ano yung mga products? Beer, no? It produces ethanol. And sa ating lactic acid, present lactic acid, okay. Lactic acid fermentation. We have milk products natin, fermented milk products, yung yogurt and cheese. Okay, dapat pa. So, that's for cellular respiration. Then we will talk about plant physiology. And to start with plant physiology, although nag-discuss na tayo katong transport, diba, last time? So sa physiology, we will talk about plant growth. Kung sa dihang plant. Naman na sa plant nutrition, di ba? And transport. Ay wala da ay nag-plant loading lang data. Tuan na po. Nawala na po ako ang transport and narang plant growth. Oh my goodness. Lagi siya ma-open. Naunsa dahil sila. Ano di man ni ma-open? One, two, hala ka, nagguba ako ang I don't know why. Nagguba akong file. Dali lang dyan ha, break sa ka. Ay, so naanan ay siya. Okay, so
Hi, this is Miss Worth, and today we're going to learn about plant hormones. Now, what is a hormone? If you took physiology, you probably learned that a hormone is some kind of chemical that is produced in one part of the body and has an effect in one or more other parts of the body. Hormones usually are very effective chemicals, and what I mean by that is that they only need to be present in very small concentrations to have an effect. And since plants don't have a nervous system, hormones are the major way that plant tissues communicate with each other. Now, the first plant hormones were discovered by botanists who were interested in tropisms. And tropisms are movements away from or towards a stimulus. And the three major tropisms seen in plants are phototropism, which is the movement of a plant towards light. Gravitropism is how a plant knows to send the roots down and the stems up. And thigmotropism, which is a plant's response to touch. Now, a long time ago, a scientist named Went, and this was in, I guess, the late 1800s, wanted to investigate phototropism, which, by the way, was a tropism that Charles Darwin was interested in as well. And what he did, he cut the tip off the tip of a growing shoot, placed it on a porous agar block, and that porous agar block acted like a sponge and sucked out any chemicals that were in the tip of that plant. Then he took that porous agar block and placed it on a plant shoot that had no tip, at which the tip had been cut off. And what he noticed was that there was some kind of chemical in that agar block which stimulated cell growth in the plant and caused the plant to grow. And that was how the first plant hormone, auxin, was discovered. So let's talk about auxin. And if you take a look at the photo in this slide, the plant on the left has been treated with extra auxin, and the plant on the right has been left alone. So what auxin is also known as IAA, or indoleacetic acid, and there's a number of different forms of auxin, IAA1, IAA2, etc. Auxin is known primarily for stimulating apical dominance, or primary growth. And what that means is that auxin will cause a plant to grow taller before it starts to branch out. And this helps the survival of the plant because the taller a plant is, the more sunlight it can get. Now, the second hormone that was discovered were cytokinins, which is also a family of hormones, also known as zeatin because the first cytokinin was discovered in pea plants, and the genus name for pea plants is Zea. Now, in this photograph, the plant on the left is the normal plant, and the plant on the right is the one that's been treated with extra cytokinins. And you'll see both these plants are the same height, but what's the difference? If you take a look at it, the plant on the right has more lateral or side branches. So what cytokinins are known for is stimulating lateral growth. Now, for a plant to be healthy, you need a balance between the auxins and the cytokinins because you want the plant to grow tall enough to get enough sunlight, but you don't want it to grow so tall that it falls over because it's too weak, and you also want it to have enough lateral branches or leaves so that it has enough leaves to capture the sunlight it needs and do enough photosynthesis. So successful plant growth is a balance between these two hormones. Another plant hormone are called gibberellins, and this again is another family of plant hormones also known as gibberellic acid, and they are also numbered GA1, GA2, etc. If you look at the photograph on left, it's showing a dwarf strawberry plant in the pot on the left, and then the dwarf strawberry plant on the right has been treated with gibberellic acid. And the gibberellic acid has stimulated stem elongation in that plant. Now, gibberellins also stimulate fruit development. In the picture of the green grapes on the right-hand side, on the left, you'll see normally sized green grapes without any chemical help. And the ones on the right, which probably they look more like the green grapes you see in your grocery store, are nice and plump and fat because they were treated with gibberellic acid during the growth process, and that has stimulated the fruit development. The next plant hormone we're going to talk about is abscisic acid. And this may sound familiar to you because we talked about abscisic acid when we did the photosynthesis chapter. Abscisic acid controls the potassium ion balance in the guard cells. What does that mean? Well, if there's more potassium ion in the guard cells, more water will rush into those guard cells to dilute out the potassium ion. That will cause the guard cells to bend, kind of like if you take a long balloon and blow it up really hard, it will start to kind of curve a little bit. and that curve in the guard cells will cause the stomata to open. So abscisic acid helps control the opening and closing of stomata. Abscisic acid is also known as ABA, and it also helps maintain dormancy. Abscisic acid is one of the chemical signals the plant will send out in the fall if it's a deciduous tree that will cause leaf drop in the plant. And dormancy helps the plant conserve its resources until growth conditions improve. Okay, the last hormone we're going to talk about today is ethylene. And ethylene is particularly interesting because it's the only plant hormone you can buy in a can. Yes, a can. Ethylene is a gaseous hormone, and what it controls is fruit ripening. Ethylene is released by ripe fruits, and this is the reason why if you have a bunch of green bananas and you want them to ripen, one of the easiest ways to do it is to put them in a brown paper bag with a banana that's already ripe. That ripe banana will be releasing ethylene, and that will cause the other bananas to ripen. This is important to farmers because if they have to ship their crops over long distances, one of the things they can do is to pick their bananas when they're not quite ripe yet, so they're harder and less likely to get damaged during the shipping process. Then when the agricultural product like the bananas reaches the destination, they can spray it with ethylene, and that will speed up the ripening so that by the time they get onto the shelves at your grocery store, they won't be green bananas anymore. Hopefully they'll be nice, ripe yellow bananas. Now, scientists for years have debated whether flowering is controlled by a plant hormone, and there's no clear answer on this. Many botanists think it is, but they've been trying for a couple of decades now to try to isolate a flowering hormone, and they haven't been able to find one. A couple things that they do know influence flowering, however, are circadian rhythms and phytochromes. Now, just like animals have a circadian rhythm, so do some plants, especially when it comes to the flowering response. Now, phytochromes are pigments in the plant, just like chlorophyll is a pigment. Some of the other pigments cause the petals of the flowers to have certain colors. And phytochromes can sense the amount of light in a certain region of the visible spectrum, and somehow that manages to send a signal to the plant to let it know when to flower. And what scientists have found out is that it's actually a critical night length that controls flowering. Now, what do I mean by that? Well, most flowering plants can be classified into one of three categories: short day plants, long day plants, or day neutral plants. Now, the day neutral plants are unaffected by the photoperiod. In other words, they're unaffected by the amount of day or daylight or night they get. Tomatoes, rice, dandelions, most weeds are completely unaffected by the length of the day and will flower as long as growth conditions are correct. Now, short day plants would be something like a poinsettia, which would naturally flower during the winter when the days are very short. What scientists have found out that actually poinsettias will flower when the days are long if you interrupt their night so that the plants are tricked into thinking the period of continuous darkness is less than 12 hours. And this is really useful to horticulturists who want to sell flowers, say, off-season. Like if you want to sell poinsettias in the middle of the summer. What you can do is you can grow them in a greenhouse where you control the length of light and darkness they get, and just make sure that halfway through the night you flash a light of the correct wavelength, and it turns out light in the red region works the best, and interrupt that night period so the plant thinks it's a short night, and that plant won't flower yet until it gets a continuous period of darkness of more than 12 hours. So short day plants are kind of a misnomer. They're actually long night plants. Conversely, a long day plant like the Marin iris actually just needs a short night. So if you were a horticulturist and you had some Marin iris plants growing in your greenhouse in the middle of winter, what you would do is flash a bright light in the middle of the night, tricking the plants into thinking that the dark period was less than 12 hours, and that would cause that plant to flower off season. Okay? Now, the pigment that detects the amount of light is called a phytochrome. And there's two different kinds of phytochromes, PR, which is red absorbing, and PFR, which absorbs far red light. And those two phytochromes can interconvert. When they're zapped with the correct type of photon of light, the electrons move around, and it can interconvert back and forth between PR and PFR. And that is thought to be the chemical that senses the amount of light that helps control flowering. Now, that being said, some botanists are still convinced that there must be some kind of other hormone that is involved in the process, but as I said before, they haven't been able to isolate it yet. So that's what you need to know about plant hormones.
Okay, so those were the phytohormones, or we call also them as hormones, plant hormones. So again, what are those plant growth regulators or hormones?
Auxin.
Yes, sir. We have auxin. What else?
Cytokinin,
Cytokinin. Very good. What else?
Gibberellins,
Ethylene.
Gibberellins,
Yes, ano yun?
Gibberellins.
Cyclic and
Gibberellins.
gibberellins. Yes. Sorry. Hindi na ako makita kasi ang ano. Okay, so to continue, this is, I just want to show you the slides kasi kanina naka-video yon. So, when they say hormones, they are also called plant growth regulators, no? Because they control growth, development, and movement. And next to this are plant adaptation. By the way, okay lang sa inyo na stay straight na na hanggang 10 to 10:30 para isa lang inyong break hanggang 1:30. Okay lang?
Yes, ma'am.
Para early mo magkadayaw, mag-indak-indak kasi wala kami nag-indak-indak to, ha? Or kamutanan unya, dili mo makagawas kung mo mag-indak-indak. No. Kasi kadayawan ka ron. So anyways, so it again came from the Greek word, what? Hormone, meaning to excite. Kasi siya ang nag-control sa growth ng tanim. Of course, review na lang ni siya, ha? So, dili pa ni first topic ninyo. And lahat ng katulad, photosynthesis, respiration, and everything, no? Formation of flowers, diba? Or even flowering, is it controlled by a specific substance or hormone based on the video presented? Yes, Bing? Yes, no? Yes, anong specific hormone ang ano ng flowering? Ethylene? Yes, no? So, but based sa ating explanation kanina, there's no such sa problema ninyo sa likod. Yes, can you share your knowledge, sir? Labong kanina, di ka katubag. Yes, pampahinog. Dili siya flower. Yes, kaya at the moment pa siya, unsa ganito, nalibog ko kasi it's not actually the one that controls flowering, ang ethylene. Pero ginagamit siya pang-induce of flowering sa mangga, sa pineapple, not really para pang-flower sa pineapple, para uniform lang ang iyang flowering. Muna na ginagamit actually ang ethylene. At saka yung in combination with potassium nitrate. No? Kasi yun yung pang-flower talaga ang potassium nitrate. Kasi ang potassium, ang function po niya is para ang mga, for ano kasi siya for, for translocation. So, madaming mga photosynthase or kanang nutrients ang ma-produce kapag higher ang potassium, kung mag-work ang potassium. Anyway, in short, it's not actually a hormone that controls or controls flowering. It's what? Kanina yung short day, long day, neutral, day neutral. So ano yon? It's the photoperiodism. It's the one, it means the amount of day and night. Kasi ang mga tanim kanina na-classify as day, short day, long day, and day neutral. Katong mga day neutral, kung itong mag-flower, wala sila pakialam, no? Whether gabi or taas ang gabi. Kasi meron tayong tinatawag na shortest, longest day of the year, di ba? And what is that? And longest night of the year. And what day is that? Burman, no? Ang longest night. Okay, so within the month of December, ano ang longest day? Hindi, longest night pa lang kasama ko, sorry. Sa December. Taysa, Mark. Nalibog na nung ko tayo sa kagaling. Pasensya na kay medyo ngarag ko. Sorry kay ganina pa siya ko. Gani. Ang mga gani ka nang hinanakit no, dili gani danon. So, move on ka ha, Dim. So, nakalimot na nung ko. I-discover ko, was it December 27 or 28? Pwede po na ninyo paki-google ko naman taekwondo ba videos ako ang online. And also there is a face-to-face. And ang ato pong longest day during summer, sir, and there is also a specific month for that. 21 ang longest day. Kuha na po ko. 9. And ang longest day, April. Okay, wala naka-stay. Kasi nga, sa atin, dito sa tropical or sa Philippines mismo, within the globe na naka-tropical, by the way, dapat kabalutan na we are in the which part of the globe or the hemisphere. Are we in the northern or the southern? Therefore, we are in the south of the equator. Makasayod, di kag paro-paro, G, sir. We are in the, sorry, sir, ha? Actually, mo na ang common nato nga knowledge, no? We are in the south of the equator, but Philippines is actually in the north of the equator. So, north tayo. Taysahay, pakita na ko sa inyo. Ay, dili April dahil, no? June 21 and then December 21 ang farm. Although wala po man na question sa board exam. Asa dahil? So, unsa ganito? Globe. Dinayo ka ba lang? Ayan. So as you can see, ang equator, ay ano man eh. Ang akong gi-share is okay. Wala din na siyang bunda. PowerPoint. Kadali lang ha kayo nakashare na. Okay, so maklaro ba? So mo ni ang ato equator, no? Asa ang Pilipinas? So we are in the north part of the equator. So we are in the northern hemisphere. So ang tawag sa northern hemisphere is the Tropic of sa taas, pinakataas, sa North Pole, of course, and this is the South Pole. So ang North Pole is the Tropic of Cancer or Capricorn? Wala pa makadungog, anak? Ay, mao ba? Sige. So these are the Tropic of Cancer, ang Northern Hemisphere. And the Tropic of Capricorn is the Southern Hemisphere. And sometimes, pwede mo tanong ka question na nganong wala kaayoy cracks sa Tropic of Cancer and Tropic of Capricorn. Kasi na sila sa pinaka-top, and then unsa may naadara mga Greenlands, diba? Savannas, diba? Well, maka-discuss mga later sa practices of reproduction tomorrow. No? So at least na-clear lang na to, ha? Kasi yes, I was also wrong last time, sir. Kabalaka. In fact, nabagsak ko ko grafting. Joke lang ito. So, ma-share ko kasi na po ay plant practices and production this afternoon. Pero, we are maybe basic sa coverage. Depende lang ha. Kaya diba, like, last time, nag-respiration pa karon in this last week, last Saturday on that to, or last Sunday to na. So, moving forward. Yeah. So, plant growth regulators is also different from animals. Kasi people and animals, we have the same kingdom. So, pareho po natin siya usually of hormones. So, when we speak of animal hormones, so ano, Sir Argel, ano yung mga hormones for animals? Katong bovine trophin hormones. Isa to siya. Ha? Unsa yung mga hormones nato? Pareha naman ta sa hayop. Pareho ra ba? Ha?
Diyan, ma'am.
Yes. Sex hormones, unsa? Kato, dili sex nga sex drive, ha? Kana. Dili nga estrogen, which is pinakontrol naman yapon niya, diba? Kanang atong testosterone, diba? So, kung pag-as kag Diyos ko, tao kaya ta ha, so dapat kabalot ta sa atong anatomy. Not only for our animals. So, testosterone is for male or female? Male. So, kung gamay kag testosterone, therefore, bayot ka. Joke lang. Ha? Wise. Wise na din mo lagay. Ala, di ako na iwan na ha. Ito dyan, Sir Dre. And, like for example, growth hormone natin sa tawag. Kalimot ko. Sa itong thyroid gland, it produces phytothyroid. Isa ito? Something na ano, no? Thyroxine, no? And that's the one which control our growth. Diba? Tangkad kay sobra na kaanin mo, ha? Tama-tama ra pod or nakulangan ng pagkaog kuan sa imong thyroid gland. In fact, growth ba or pagkapayat or kataba? And also, so those are usually hormones. And mamaya sa ating plant breeding and genetics, we will talk about more on organisms, mga genetically modified organisms na controlled by hormones na. Katong mga GMO hormones na. Like, pwede na ka makaproduce og manok in just 2 weeks, diba? Kadungog na mo, Sir Lags? Okay, sa imuhang kuan, sa imuhang dogs, sa imuhang usually nag-breed man ka, diba, Sir Lance? So, ha? Unsa imuhang new knowledge naman i-share sa mga in your personal experience? Sa pag-breed gani og dog, alangan naman sa ha? Selective breeding. Then unsa may dapat ipakaon ana nila para 100% gid ang kanang chance of winning sa breeding? Nay mga technique dira? Or ha? So kung bitukon siya, dili siya kay makabuntis. Really? Mo ba? Tama ka? Sure na ka, ana? Sure, jud ka? Practice or knowledge? By practice. Nakita na ni mo ringworm sa sulod. So, practice niyo nagadi-worm mo before breeding. So, kanang dili mga buntis dira, mag-deworm pag ka, ha? Okay. So, for plants naman, it's just different, no? Like, for example, plant hormones are not made of tissue specialists in hormone production. Like sa sex hormones in the gonads sa humans, in the pituitary gland, sa brain. So, namaligay ko ganina, ang gatong thyroid gland, pituitary gland kasi ang sex hormones, ang thyroid gland sa, okay, tama din akong ingon, ang sa growth o kataba. So, in plants naman, again, for example, we have auxin na mag-stimulate sa, like for example, sa roots, no? Common man yung auxin is for the root growth. And that's why sa mga nursery, mga nursery operators dire, or nag-work sa nursery, nag-apply ba taog ka ng anaa or alpha-naphthalene acetic acid? Kung sa cuttings na to, yes, Sir Dre. So, Na is naphthalene acetic. Unsa pa? Sa tissue culture, sa yung gamit? Sa ano? Mag-routing na mo na wala mo ikuan abscisic acid, cytokinin. Wala, but during multiplication stage siguro. Okay, so anyway, kanina slide is just presented already from the video that we just have shown. No? So, pwede siyang endogenous, exogenous, ibig sabihin, there are already normal hormones inside the plants. But then why do we have to apply pa yung in-apply nila, Sir Dre, doon sa kanilang nursery, sa tissue culture, and also sa mga cuttings? And yeah, bakit kailangan natin o synthetic substance pa? Para mapadali, alter their growth, mas mapadali pa din siya. So it's again a natural substance already, but they are in lower concentration, as in micromolar sila or less pa, and it can easily be translocated and ma-dissolve or ma-react, responsive kasi sila. So pwede sila ma-in conjugation, no, pwede siya mawala, daghan mahitabo kasi gamay lang. So kanang maayo susabing, no, gamay-gamay lang ta pero daghay mahitabo sa tuwa. So dili sila small but terrible, tingnan mo dyan na siya. Okay, mawag siya mo control. So, if we need, that's why we need to supplement them in synthetic amounts. So these are our 5 major hormones for plants. So dapat memorize na ninyo kalimaran na kabuok, no? Auxin, cytokinin, gibberellin, abscisic acid, and ethylene. So isa-isa ko muna nato, and we have also newly discovered hasmonic acid. Para nag-control po na siya sa stress sa panom, racinosteroids, salicylic acid, and polyamides. And not yet confirmed peptide hormones, oligosaccharides, phospholipids, and the mRNA or protein florigen. Kasi they are still, as I have said, wala pa na prove na mag-control sa flower, hormone na mag-control sa flowering. So they are proposing florigen or for floral induction. But then, wala pa sila complete evidence or explanation on that. So, to wrap up the function of the major plant hormones, when we say cell elongation, auxin na siya. So, mo yun na yung main purpose. That's why pwede siya pang routine. Gibberellins naman is both for cell elongation and division. That's why daghan ka na siya ginagamit. As well as cytokinins for cell division, and it will inhibit senescence. So, dati na-remember na ako sa UP, experiment me o ganang leaf cuttings. Ibuta na mo sa water only, and then petri plates with water only, and plate with cytokinin. So, after a week, ang katong water only layak na. Pero katong with cytokinin, sa color. Green pagyapon siya after 1 week. So, for abscisic acid naman, ito yung hormone, that's why ma-abscise, matanggal atong mga leaves and fruits. In fact, ginagamit na nila na chemical kanang thinner sa ubang country like sa apple. Kasi para dagko kayo ang kuan sa apple, ang ilang prutas, gina-thinning na nila. Then ginasprayhan lang of abscisic acid. So, yes. Ay, tangi. Sorry. Ano? Na-share na?
Yes, ma'am.
Sorry, Sir Ron. Actually, ang amo na lang gikuan kay na dugay man may istorya sa animal god. So kanira man, the major hormones. And of course, don't forget na it came from the Greek word hormone, which means to excite. Then it could be endogenous or exogenous. And yeah, low concentration. These are the 5, again, major hormones. And the secondary, newly discovered. And others for confirmation. So again, these are the 5 major hormones and their general function. Lastly, we have ethylene, which promotes senescence or matigulang siya. Epenastic, kanang mabawog siya. Kana ka-remember ka, agakuan mo anak, or one by one man ni siya makita ninyo. And also for fruit ripening, but not for flowering. But then, it is also applied for uniform flowering. So, auxin, daghan kayo siya o ganang infos, no? Kasi, ang auxin kasi ang first na na-discovered, kung ka-remember mo sa video, si Gwen, nag-slice o katong tip, ito na yung mga bloke-bloke kanina. So si Gwen, itong scientist, naka-discover na na-aday substance or hormone ang plants na mag-control sa growth. And then first, na-discovered ang auxin. And then second, ang gibberellic acid, then cytokinin, and last na ang ethylene. May gani, wala sila i-discover, no? Ganang kitag studyhan. So, bitaw. But then, we are still continue with new knowledge and also research. Kasi even yung, hindi pa nga proven yung florigen, no? The supposed hormone to control flowering. Yan. So, ang effect ng auxin is depending sa iyahang concentration. Although they occur only in very low concentration, but kung sobraan po ng auxin, kung gamay lang ang auxin, di ba? And then level po na sila sa cytokinin para mag-control ang iyahang routine. Otherwise, kung mas gamay ra imong auxin, mas mag-grow ang shoots. No? But kung too much with auxin, natay mga ginatawag na 2,4-D or DDT, Agent Orange. Ma-mention man na siya doon. And it increases the plasticity of the plant cell wall in stem elongation. So mo niya ang experiment ni Wen, nganong na-prove niya nga na nag-control sa growth, kay ngano, pag itabunan niya ang tip, unsay na itabo. Wala siya, ang normal is mabawag siya towards light. But katong tanggal niya ang tip, wala siya nabawag, diba? Itabunan na po niya, mura lang siya condom, wala siya nabawag. Pero, transparent iyang gibutang, nabawag siya. And very ang gibutangan niya sa baba, nabawag yapon siya, no? And I-block the rate, nabawag siya. Therefore, nagit sa tip. Diba? Nadiri, therefore, ang auxin na nag-control sa growth. And another experiment to prove that, asymmetrical placement. So, and so on, na-prove nila na using the coleoptile of in gelatin. So, then another experiment, nag-apply na din nila ang auxin, then dito na din nila nakita na muna yung mga, pag magbawag siya, the more ang auxin. Kasi overgrowth na rin, di ba? Kanina, elongation of cell ang auxin. So, that is just to demonstrate lang ito sa experiment. Ha? And these are the natural occurring auxins. We have indole 3 acetic acid, chloroindole 3, phenylacetic acid. And these are the others, the 2,4-D or mo ni ginagamit na herbicide. Kasi ingon gani ko ganina, pag sobra siya kadaghan, nakapatayin po siya o sagot. This is the naphthalene acetic acid, the dicamba, and the picloram. So, meron silang aromatic and carboxylic acid. So, this is very, physiology. Yan sa nila pag, they came from, yun sa sila pag-produce sa tryptophan pathway during synthesis. And they usually found in young developing leaves, buds, seeds, and they are transported basipital or away from the tip, apoplastic or outside the cells, symplastic or through the cells in plasmodesmata, and phloem conjugated form or glycosides in glycosides. So this is the tryptophan dependent pathway. So ganina sa respiration, bali mag-link na lang ni sa respiration. So, and this is how the auxin polar transport. Dili na kailangan ninyo sabton, no? Ako lang ipakita. And what are the mga importante na dapat niyo timanan? Unsay mga effects sa auxin? So, dapat you understand that it can enlarge the cell and also able to increase shoot growth through the internal tubers and valves. and root growth in the storage roots and adventitious, and fruit growth sa strawberry. So receptacle enlargement. So auxin, if this is also an experiment to show you that it will elongate. Okay, so ito ang effect ng auxin. And water lang siya ang letter A, and auxin ang letter B. So, mas tagas kayo ang auxin. And for the, actually strawberry is not a berry, ha? It's an aggregate fruit. Ang true fruit niya is actually this one, the auxin. This is just a modified or swollen receptacle. So kung i-remove niya ang fruit or auxins and they sprayed with auxin, and kani there is the normal fruit, so nindako gyapon no ang prutas ay ang strawberry. Maski pag wala siya auxins but gisprayan siya auxin. And auxin is also for apical dominance, meaning mutaas ang imong fruit kung meaning mo putan of it. A fruit, I mean shoot. Like anything, no? Like an Indian tree. No? Pataas siya kung because of apical dominance. Then the ratio of auxin, pag high, maging dormant yung axillary bud, kasi kaya nga pwede siyang herbicide. Pag low naman siya, it will enhance growth of its axillary bud. And it can also control adhesion, no? Kung maggamay na lang ang auxin na daghan ng ang ethylene, therefore matanggal na siya, semi-sim. So, maski pag nag-yellow na labiha, pero daghan pa kay kag-spray ng auxin na dara, dili na siya matanggal. So, inaana siya pa, So, this is apical dominance. Once you don't cut this one, it will continue grow upward. So there are a lot of mechanism of action for auxin, no, for cell elongation. Sometimes these are asked in the board exam. By this through bind receptor protein plasma membrane, and it will transport into the cell in the activating ATPase and hydrogen ion exclusion, and it will acidify the cell wall, break hemocellulose pectin bands, and so wala siya ginatawag na cellular lang siya. Kasi sometimes ang pangutana last time in my time is which among the different hormones have genetic the mode of action is genetic, no? So, but for auxin, it is not, it has no genetic modification na function, cellular lang na mode of action. So, this is the mode of action in loosening the cell wall. No? Grabe kaya mga scientists na nag-propose aning a theory. Signal transduction pathway, wherein ang auxin daw mag-interact siya sa mga calcium ions to make it into calmodulin. Is a protein na makaregulate sa mga processes sa plants, animals, and microbes. So, muna iyahang signal transduction pathway. For uses naman, rooting, propagation, no, natin yung tawag ng hormone, rutone, commercial preparations ng 2,4-D. As a herbicide in higher concentration, ang 2,4-D makapatay siya sa dicot but not so much sa monocot. So, dicots have 2 cotyledons sa kanang mga dagko nga leaves, broad leaves. Monocots are the katong mga leaves lang na venated or na pareha sa corn, in short. With control in cereal production also. Like the Agent Orange, no, in Vietnam War, kasi dati mga wais mo ng mga Vietnamese, diba? And they're so proud. Until now, nawala sila na pildi sa Americans, diba? Sila lang the only country from Asia na wala na colonized by Americans. So, Huwag na kayo sila matago. And kasi di man nila makita ang mga Vietnamese soldiers. So, sa liwa sa mga Americans, disprayan nila og 2,4-D and 2,4,5-D para di pulitan ng trees, di na makatago ang mga Vietnamese. And then, but now it is banned. Prevent also abscission of leaves and fruit sa older leaves, ripe fruit, indigenous production of IA stocks and replace na siya by exogenous na. So that this is also the reason why kung kailangan to mag-spray of hormones and use for chemical cleaning as I have said earlier. In tissue culture, kung high ang auxin, mag-root siya. Pag low, mag-shoot siya. Kung equal na ang ratio sa auxin and cytokinin, callus formation will occur. And it will promote also flowering and fruiting. And for sex determination, no? For development ng apical dominance. Sa cucumber, kung mag-spray kang auxin, mas daghan siya og male fruit. So this is the cytokinin, zeatin, and the isopentyl adenosine, the IPA. Yung rising drawing, ha? So it was discovered by Haberland in 1913, and later pa, dagan pang mga na-discover na mga cytokinin. Like Miller. Yan ako isa-isahon, ha? And these are the natural cytokinins. The kinetin, the dehydrogenases, and the zeatin. And we have the synthetics, the backbone yung ginagamit ni Sir Dre, benzyl aminopurine. So, ang gamit ng cytokinin, although it's already found in angiosperms, gymnosperm, and all plants, in the root, seeds, fruits, and young leaves. So, if ang question is asa makita ang mga hormones, dapat kabalo mo kung asa makita, ha? And it's synthesized in the mevalonic pathway. Kasi yung kanina, ang auxin is synthesized in what pathway? Kanina? In the, ang cytokinin ang mevalonic, ang auxin, dito, tryptophan pathway. So, ang transport naman ng katong ganina, ang auxin, yung mga basipital, lateral, for cytokinin, upward and silent, non-polarly and silent lower right cells. So, these are the different pathway for plant, for bacteria of the cytokinin. Just to show you. So, anong gamit ng cytokinin? Para sa cell division. That's why ginagamit siya tissue culture para the more shoots and the more proliferation. Morphogenesis, lateral bud development, and nakadelay and senescence or paglaya. Apical dominance. No? Pag high ang auxin and shoot uptakes, ang cytokinin ang for apical dominance siya. And if high ang cytokinin root uptakes. And the concentration, pag high ang auxin to cytokinin, dormant lang ang mga buds, but pag low ang auxin, pas high ang cytokinin, mag-branch out siya. So for shooting and branching out po siya. For example, the crown gall cells have acquired a gene for cytokinin synthesis. So therefore, klaro na diritaan, no? Na if there are questions like which hormones which can alter or kanang something genetic modification ang iyang function or mode of action, so cytokinin is one of them, the hormone, but not auxin. So kasi siya o tingnan mo sa crown gall, no, proliferation, and it promotes chloroplast development, no. As you can see, promote movement of nutrients. Without cytokinin, dili mo dagan ang ang kanang element or nutrients. And ang chloroplast food for green. So this is the model of the mode of action and signaling of cytokine. Anong gamit niya sa roses and chrysanthemum for axillary buds induction, micropropagation, and tissue culture? Yan. So organogenesis ang tawag niya sa proliferation. Again, pag high ang cytokinin, unsay mo form? And pag low ang cytokinin, taas ang auxin, unsay mo form? Good. Next, we have GA. So ito yung, it was actually named after Gibberella fujikuroi. It's a fungus na makakos ng abnormal tallness sa rice. Kanong mo, anak? Tangkad kaya ang rice. So pwede siyang active or inactive phase. GA20 and GA1. So we have 50 more, 100 more gibberellic acids. No, sa cytokinin, sa auxin kanina, natay NAA, DDT, diba? Pero sa GA, ang tawag sa iya GA20, GA1 hanggang 50. So this is when it was discovered in 1930 for Bacane or foolish seedling disease of rice or the Gibberella fujikuroi. So Kurosawa, ang scientist na nag-name ang gibberellin. So how does it synthesize in the carotenoid pathway? So again, ang auxin na synthesize, asa siya gikan ba? Yun sa niya pag-create through the what pathway? The Tryptophan pathway. Ang cytokinin, mevalonic pathway. Therefore, ang gibberellin sa carotenoid pathway. In the, where exactly? In the endoplasmic reticulum and cytoplasm. So, asa niyo mo siya makita? In the shoot, apical meristem, young leaves, apical internodes, developing fruit, germinating seed roots, and stem. So most parts. And it can be transported in the xylem phloem through nonpolar or bidirectional. Maski yung saan siya, no, depende sa iyahang mga kuhan. So these are the stages of the biosynthesis in the carotenoid pathway. Ayun na siya memorize, ha? So, unsay gamit sa GA? It's for elongation. It will induce division, elongation, and it can also break dormancy in buds and seeds. No? That's why muna siya ang main hormone na maka-germinate sa seed. Kasi normally, ang seed, na sila ay abscisic acid. Ang abscisic acid, remember, it will make the seeds dormant. Then, pag ma-activate na galing si GA, so mag-germinate na siya. Especially in cereals, barley, but not so much in daikon. And it can also, again, promote the flowering of brassicacea. Kung spray niyo muna siya, ma-enhance ang flowering. No? Instead that it will take 2 years para mag-flower ang mga cabbages and so on in its family, pwede nilang mo sprayhan og auxin. Ay, check, gibberellin, GA. So, it can make also your plant dwarf. Kaya ginagamit siyang pang-dwarfing root stock sa apple, pear species, no? And fruit quality from scion. Ayan. And also, it makes the seeds parthenocarpic or seedless. I mean the fruit, parthenocarpic or seedless, like the Thompson seedless grapes. Okay, imagine, this is the normal grapes. Pag spray mo GA o bongga, siya diba, mura na siya bouquet. And also for seed germination, sa malting barley, precocious germination. Therefore, kita na mo anang kanang mais, kakao nga nag-germinate sa sulod pa sa prutas. Na siya term, which will be mentioned in the next slide. Male flower production din ang GA, no? Sa mga monoecious and dioecious plants, don't worry. Mag-mention pa taon sa mga monoecious and dioecious. Pag monoecious siya, lalaki lang siya or babae siya. Pag dioecious, well, pwede siya any, no? Dili, I mean, for flowers, ay kung monoecious siya, the same plant na siya babae na lalaki. Like the cucumber, diba? And also, hello? Asa na ka? Ano na lang sa RMCP Tanite? Ikaw akong paleksyon? At sundo ako RMC para bongga. Sige na. Uy, nag-reviewer pa ko. Nag-review pa ko. And mabot ka din mo, undang na ako, promise. Sunduan ako din. Basta sige ka. Ha? Basta ka RMC? Asa mo kayo masali ka? Ay, di man, abrisa lang ka. Kasi gawin na kita na sa abrisa. Mano na ako eh. Sige. Okay na. Okay, sorry ha. So, yeah, for GA, and dioecious is that babae siya separate, lalaki siya separate, like papaya, diba? Like babae yung lalaki nga kapayas na punuan. Kabalungwana, diba? Sa pagkabalo kung lalaki o babae, ang kapayas.
Dili mamunga.
Yes. But mubulak sila, both na sila flower. And then, dili lang ma-form ang lalaki o prutas. Only the girls, the babae, of course. No? But later, we will have distinction kasi there na po yung mga whatever. Na mga types of flowers. No, ma-discuss na po. Ano naman yung ako ang kuan? And then, for chilling requirement, no, para di na nimo kailangan ng freezer, pwede sprayhan na lang nimo o GA. So, the Samsung seedless grades. Rosetting or bolting na for GA. Ayan, nag-flower na siya for year 2. And for serial seed germination, in the figure, it just means that the GA are synthesized in the embryo, and then it will be absorbed through the scutellum, and siya ang makapa-activate sa enzyme na maka-degrade sa starch. And this is how GA works. And these are example, the abscisic acid na ta. For abscisic acid, we have ABA or xanthocene. So how does it discovered in 1940? Kasi they were searching for hormones na maka-inhibit sa growth, and they call it Hemberg, called it dormins. And then later na-discover na yun siya. And it is synthesized where? In the chloroplast sa mga mature green leaves. Through what pathway? Terpenoid neverone. Ha? Ano sa daw? Ay, ako to. Oh my God. Ugma pa ang kwan sa ko. And duha yang ways ha, carotenoids, biolaxanthin. And it is transported in the tissues non-polar. So this is the biosynthesis regulation in the terminal pathway. Ano ang gamit ng abscisic acid? Retardant siya. No, dormancy and drought resistance. And ginagamit siya sa post-harvest storage. Kasi maka-dormant siya eh. And this is what I said, no, vivipary. Pag lack ng abscisic acid, so ang GA na lang ang mabili and precocious yan, germination, nanubra. Wala pa ganun siya na-harvest, nag-germinate na siya. And GA in stomata closing and opening. It's the abscisic acid na wrong na siya. Kasi dapat ABA ni siya. So it's the abscisic acid ang nag-control sa pag-close o open sa stomata. And of course, with the reaction ng mga calcium ions and potassium ions. Pag ang potassium mo gawas, mo close ang stomata. And pag potassium mo sulod, mag-open ang ABA will inhibits the stomatal opening. So this is the simplified model for the signaling ng yung stomatal opening. So lastly, ethylene. So that's C2H2. It's colorless. It's a gas and it's produced in all plant tissues kapag stress, ripe na siya or senescing. And it's synthesized in the methionine or the shoot apical meristem. So this is the pathway in the young cycle. It's the methionine pathway. So how does it discovered in the 1800s? Kung katong nagsunog ang gas and then ang sa inahitabo, na-develop ang short stick stems because ang yung mga limbs na fall off. And so, later on, na-prove nila. So, ang function niya for fruit ripening, epinastic, downward, curvature of the leaves, break seed and bud dormancy, induces formation of roots and hairs, and flowering in pineapple, bromelias, and mango. Wala ko nag-delete. It will also encourage senescence, abscission, initiation of stem elongation, lateral cell expansion, and sex expression. So, maka-female siya. Kung ang auxin makapalaki, ang ethylene makababae siya sa cucumber. Okay, so this is the theory how auxin control and ethylene control leaf excision. So, pag ang auxin gamay na lang, ang ethylene mugawas siya ang makapa-fall down. So, for uses, for fruit ripening again, fruit ripening, and floral development, sex expression, and the greening ng citrus. It will not make your citrus ripe, but it will just make it orange or yellow because it will break down the chlorophyll. And mechanical harvester, post-harvest shelf life. I-inhibit lang niya through silver thiosulfate. And through ethylene inhibitor, siya lang ang ginapulperya para ma-increase ang shelf life sa using irradiation, carbon modified controlled atmosphere, or kung low ang temperature, wala na dito ng aircon, actually aircon, wala lang siya na-close. I-on na mamaya. And the 1-MCP money study ni Sir JP, no? Nagamit siya MCP para ma-increase ang greenness sa Cavendish ata to. Okay. And other hormones maybe we'll discuss is gamay na lang man yun, 84. Hapit na lang, humano na lang na ako. So katong other hormones ganina, ang jasmonates, pang senescence siya during stress condition. Wound responses. So, pag stress ang tanom, muna ang mga hormones na mag-activate para makakoop up ang tanom with stress. And also, brasinosteroids, no? Steroid naman siya, growth promoter from grains brassiconapos, tea bean rice, and for apical dominance senescence. It will also inhibit root growth and prevent premature abscission of roots. So, daghan siya na-discover ng mga ano. So, nakatulog na mo brassinosteroids. Okay, muna yung function. Elastic polyamines and salicylic acid. No, it will promote tissue, stabilize membranes, minimize water stress, and delay senescence. For salicylic acid naman, it will also enhance male flowers and induce resistance to disease of plant. So we have 3 hormones na which can influence the sex character of plants. No, we have auxin to make it male, ethylene to make it, and we have salicylic acid to make it male. Okay. So, meron din tayong synthetic growth retardants para putot gyapon siya, gwapo siya tanawin, no? Like the diaminozide, the chlormiquat, kadong mo ano nila? Sumuna yung mga gamit. And lastly, the ansimidol and paclobutrazole. So, paclobutrazole is my favorite. Ginagamit, nag-study ko sa iya twice for the enhancement of flowering ng citrus or ng pomelo. And okay, so questions so far. Clarification. So medyo na-focus lang dito sa mga auxin, although nakaka-type topics. But I promise you kasi na mag-11 to 1:30 ka, no? Or 10:30 para ano. Ay, how about sa online? Do you have questions or clarification?
None, ma'am.
Wala. So it's been a long discussion, although 5 lang siya ka auxin. Ay, hawad kay si Nova, okay? So according to Miss Nova, yung mga kanina na hormones for animals, we have estrogen, progesterone, and testosterone. No? May na-explain ba na ni Sir Vincent sa inyo? Mamput kayo na magawas good. Ang usually nagagawa sa animal science, kay katong mga gano'n siya manganak, gano'n siya dapat siya ipaupa para mabuntis. No? Tagaan mo, situational nga. Karon kay pwede na siya, inhit na siya. So, mga inanak. So, gano'n siya manganak if mabuntis siya. So kabalad, di ba ang swine is 3 months and 3 days. 3 weeks and 3 days. Okay, so far break na lang muna tayo hanggang I'll see you at 1:30. Okay lang ba sa inyo or gusto din niyo mabalik agad? Okay, sorry ha, kay kung ano. Ha? Yes, Bim? Gusto niyo mag-activity na dahil takaroon? Para makasayaw na mo tanan? Ha? Sige, by group na ta ha. So we have the group Auxin, the group Cytokinins, and the group Gibberellins. So kung mag-mention na yung function, kailangan mo tingnan mo Auxin. No, Auxin. Or Cytokinins, Gibberellins. No? Kung di mo gamit makatingin ka rin sabay-sabay, let's forget ako sa function. Therefore, manayaw ka na. Manayaw na yapon ka na. Okay, so ready? Kapit mo sa online. So it is for sex determination. Uy, Auxin, pildinan mo. Sige, tawagan sa nato, be. Asa mga Auxin? Ingon tayo nyo, Auxin. Tindog dayon or istorya na lang. Auxin, no? Okay. Cell division. Ang Auxin po kaya, no? Elongation na ba? Wala division. Ay, sorry. Ang Gibberellin code. So, dapat duha mo nagtingog. Diba? Again. For chemical thinning. Uy, Auxin. Liman, kanta di mo manawag. Okay, for tissue culture. Cytokinins. Listen siya. Okay, kani, rooting, hormone. Oh, very good. Okay, ni, for ripen, ay, wala na yakin. For seed germination. Ay, otro po ning Gibberellin ay. O de, makasaya yung mutanan o ay, sample pa to. Dagan na kayo mag-violations. So, i-continue na nakaratot karang hapon na ganang function sa Auxin group. So, mamaya, ang kuha na po, pilangon na to ang ethylene o ang CC. Okay. So, wala na tayo pangutana para maka-break na ta. So, everybody stand up for our closing dance. Really closing prayer and closing dance. O, di. Tuloy lang, ha? Uy, unsay tugtugan ninyo? Bim? Ha? Dili karon. Tama na ako ang spelling? Ang sana na pagsayaw. Sige. Freestyle na siya, freestyle. Hindi ka uli, ang hindi mo ang sayaw. Uy, nakita ka ron. Wala, hindi ka uli. Hindi, freestyle. Kay Bim, masutungan niya si Bim. Dali, Bim, dali ka, Bim. Saan ka ba? Wala, digpin mo ka ulit. Wala, gani. Gusto ka tayo na, bacon na lang sayaw? Si Bim, oh, nana. Bim, dali ba? Dali ka ba? Ana-ana raon sa day mo. Wala lagi sila ay. Ah, sige, Ben. Pwede na mo ulit si Ben. Oh, assignment na ninyo niya ha. Thank you very much everyone. Kasi di na mo mabalik ko niya ha. So we will see you at 1:30. Okay na? 2:30? Okay na? 1:30 p.m. Okay na? Magkita ka 1:30, okay na? Exactly 1 or 12? So, we'll have an early lunch break lang kasi nga kadayawan. So, see you at 1:30 po, mga online.
Thank you, ma'am.